In this module, we will look at the organization of the auditory system. We will start off by understanding sound and how that travels through the air. And then we'll look at the structure of the outer ear, making our way to the inner ear and eventually to the cerebral cortex. Please keep in mind the hierarchical organization discussed in the previous module. Contrary to eyes that respond by photons that are impacting the photoreceptor cells in our eyes, the auditory system works by sound, which are nothing more than vibrations of air molecules. The three physical dimensions that we look at to understand sound are amplitude, frequency, and complexity. By amplitude, we know that we're looking at how loud specific sound is. If it's a loud sound, we would say that there's a high amplitude, and that is to say that the characteristics of the physical stimulus would be higher waves and deeper valleys. And we're talking about something with a little bit of less decibel output, meaning not as loud. We would say that there's a lower amplitude, and you would see lower peaks and lower valleys. Frequency is measured in a similar fashion. However, frequency looks at the pitch. That is to say, whether you have a deep voice or whether you have a high pitch voice. Think of Barry White, one of the most famous singers with a very, very deep voice. The frequency would actually be low. And the characteristic of a low voice or a low frequency sound is that there is more space in between each wave per second. When you look at a high pitch or a high frequency sound, you see that there's less space in between each wave. An example of a high pitch or high frequency sound would be the voice of Mariah Carey. Complexity is the third physical dimension, and this is how this is perceived by slightly different waves. Notice how these seem to be a little bit more smooth across the board. This seems to be interrupted by different things, and the way we perceive complexity is to what we call the timbre. And this is the character or quality of a musical sound or voice that would be distinct from its frequency and amplitude. So when we look at the timbre, we're talking about a pure sound, which would be a little bit more along the lines of a steady, versus a rich sound, which would be a lot more complex in the way the sound would be perceived. Please note that natural sounds are complex patterns of vibrations because there is a lot of movement and a lot of other noises that would be sensed along the way. If you want to have a pure sound, you would need to get yourself into a studio where there is a lot of protection from outside world, and we would not be able to then sense or perceive the rich areas of the sound or the timbre. In this picture, we can appreciate the breaking down of a sound. Please note that looking back at the complexity, the physical dimension of a sound, here we're able to see the sound, the breaking down of a sound, in this case the sound of a clarinet, into its component of what we call the sine waves by an analysis. Here you can see that an individual that would be playing the clarinet would have a range of noise of different amplitude and different frequency as well as different complexity. However, when added together, you end up with a sine wave that would produce something along the lines of this. And this would be a wave form of a clarinet sound. We understand that there is some sound of air embedded within, but what you and I are able to perceive is again the timbre and as well as the frequency and the pitch. There are some that have argued that amplitude and frequency in individuals who are able to hear would be natural and that all of us would be able to perceive and sense that. But complexity may require an experienced musician or an individual who is able to tell perhaps when there is a change in a note, when there is a string that is flat or a string that would be sharp. So while some argue that complexity would be more of an innate or a learned perceptual dimension. There are some that still believe that we are born with a capacity but that we may just not have exercised it enough. In order for us to be able to hear, the sound waves must enter the auditory canal of the ear. There they will cause the tympanic membrane, also known as the eardrum, to vibrate. Looking at the outer ear, please note and notice the area and how the ear is shaped. As the sound waves travel, please note that they're impacting this area here, and if you will, they are trapped by the shape of the outer ear, funneling, if you will, the sound waves so they could then impact here. From there, they will start generating vibrations in the three ossicles, which is what we call the three little bones in the middle ear. These trigger vibrations into the oval window. Now please note that the ossicles that we have in here are what we call the malleus, the incus, and the stapes with this one here. Here you can see the spelling of these names. Once again, as sound wave enters the outer ear, it goes through the ear canal, and then it causes the tympanic membrane to vibrate, which will then cause a domino effect of the malleus vibrating, which would cause the incus to vibrate and the stapes to vibrate. Once it enters the oval window, the vibration of the oval window would set in motion the fluid that is in the cochlea. The cochlea is this snail-like thing here, and you could see that if we unwind it, it would look like a straight component like this. If we were to do a cross-section of the cochlea, that is to cut it right here, this is what the inside would look like. Please note that inside the cochlea we have fluid that's just constantly sloshing back and forth. Those vibrations that were generated by sound waves would actually set in motion the fluid of the cochlea, and the cochlea's internal membrane, which is what we call the organ of Corti, would be the auditory receptor of the organ. The Corti is composed of what we call the basilar membrane. The basilar membrane would be this component here. Right at the dorsal part of the basilar membrane, we have what are the hair cells. They're mounted on there, and they're protected by the tectorial membrane, which is right on top of this right here. The stimulation of the hair cells would trigger action potentials in the auditory nerve, which you can appreciate and see here in a light blue color. Here you can see a different picture with the wound up cochlea, whereas where the hair cells move in the basilar membrane, they would then trigger an action potential, and then they would synapse over to the auditory nerve, which would start making its way over to the brain. Similar to how the visual system was laid out in a topological fashion, in what we called retinotopic, the ear and the auditory system is also laid out and organized in a tonotopic frequency. By tonotopic, we mean, again, that there's a mapping, a component that allows us to be able to locate the parts of the cerebral cortex that would be stimulated given specific sounds. Research indicates that higher frequencies produce greater activation closer to the windows of the basilar membrane. That is to say, remember, keep in mind that the basilar membrane is the entire thing from here to here all throughout the cochlea. So the basilar membrane window would actually be somewhere toward the beginning part and in the area here. However, lower frequencies, and that is to say by frequency we're talking about pitch. Keep in mind that higher frequency would be closer to here and would impact the basilar membrane at a closer range. Lower frequency, that is to say a deep voice, would produce greater activation at the tip of the basilar membrane, and that is to say all the way up on top. So we think that the vibration, depending on the frequency, would either travel all the way up on top and activate more parts over here than higher frequency, which would activate more parts all throughout the entire membrane. contrary to the very complex pathway that we find in the visual system, from the retina to the geniculate and all the way to the striate, there is no major auditory pathway that's similar to that. Instead, there is a network of auditory pathways. And so here in this image, you can see the pathways. Now notice that the signals from each ear are combined at a very low level in the superior olives, and then they're transmitted to both ipsilateral and contralateral auditory cortex. You may remember that ipsilateral basically means it stays on the same side. Contralateral, it means that it crosses over to the other side. Having received the information from the basilar membrane here inside the cochlea, and with those hair cells moving, it would activate an action potential that would then synapse over to the auditory nerve. Notice here that from the auditory nerve, it would synapse over to the cochlear nuclei that are in the respective side of the ear. In this case, we're looking at the right ear synapsing ipsilaterally to the cochlear nuclei. From there are various projections that would lead over to the superior olives that are ventral, right? Ventral toward this right here, and anterior. So you'd see that it would go from here to some of the superior olives, but there's also connections and that are made in a contralateral fashion. From here, the axons would then project via this nerve right here, the lateral lemniscus, and that would then go over to what we call the inferior colliculi. Colliculus for singular or colliculi for plural. The axons from there would project over to the MGN, and that is the medial geniculate nucleus of the thalamus. Remember that the lateral part of the thalamus is reserved for visual. The medial part, which is closer in, would be reserved for auditory. From there, it would then synapse over to the primary auditory cortex, and then we'll talk about the cortex distribution in upcoming slides. Please note that we're still focusing on the subcortical mechanisms of sound location. By subcortical, we mean underneath the cerebral cortex. When we talk about cortical surface, then we're talking about the top of the cerebral cortex. In this case, since we're dealing with the midbrain and hindbrain, we know that it is underneath it. A little bit more about the superior olives, which are located here in the hindbrain. We know that each of these react differently to what is heard by the ears. What we know is that the medial part of the superior olives is known for its reaction to arrival time differences. That is to say that if you were to look at a discrepancy in a video between the movement of the mouth and then the actual speech, this is the part that would somehow send a message to alert you that there are differences as to how the information is being received. We also know that this can also be seen experienced without visual information. So instead of looking at a video, perhaps if you're listening to a song that carries a tune and you're expecting a particular pattern and the delivery of a note every so often, the absence or disturbance of that timing would also be perceived by the medial superior olives. The lateral superior olives, which would be on the side, these are known for amplitude differences. So if you are able to hear in a lecture professors that do a good job of regulating their voice when they want you to know certain things, they may, instead of speaking in a monotone voice, they may actually just increase their volume when something is important. And you would then be able to sense and perceive the difference, and that would activate this lateral superior olives. From there, they would both project over to the inferior colliculi. Please note that I made a mistake here, and I unintentionally wrote the superior colliculi here, and I kept writing that, so I've made the correction there, and I've written that I meant inferior. Please note that again, from the superior olives, depending on what you heard, we would synapse over to the inferior colliculi in the midbrain. The deep layers of the inferior colliculi are laid out according to the auditory space. That basically allows location of sound sources in the world. You may remember that the LGN has different layers that are responsive to movement or the details. Think of it this way. The deeper layers in the inferior colliculi are laid out according to auditory space, and they dictate where the sound may be coming from. The shallow layers of the inferior colliculi are laid out in what we call a retinotopic fashion. That is to say that they are making use of the visual system. The benefits of understanding this complex system, auditory system, are many. In 2014, a paper was presented at the Computer Music Association conference, and they captured the attention of a lot of advertising experts. A PhD student authored a report that found that TV commercial viewers have much better recall when the soundtrack is slightly out of sync with the video. The adjustments of the timing were imperceptible to the eyes and ear, but the results were dramatic. The concept of mis-syncing the audio, which is to not be in sync, is a form of dynamic attending, which tricks the brain into anticipating what's next when it hears the audio before seeing the video. This anticipation causes the brain to remember the information much better. Interestingly, this type of information would have only been acquired by individuals that are doing research with and that understand the layout of the auditory system, both from the outside and inside. After traveling from the outer ear through the tympanic membrane, the basilar membrane, through the hindbrain, midbrain, the MGN, we finally make it to the cortex. The auditory cortex is located in the temporal lobe, and you can see it here. Please note that the core region includes the primary cortex, and that is basically inside. You can see the arrow. We would not be able to see it subcortically. We would actually have to go inside and be able to feel if you were to put your hands on this in here. The belt is what surrounds the core region, and this secondary cortex, which is the lighter shade of pink in here, would be referred to as the parabelt area. Please note this is very close in some way to the occipital lobe, and we will see how there's a relationship or can be a relationship between the two. Recent progress of the study of human auditory cortex has resulted from the convergence of functional brain imaging techniques, meaning fMRI in humans, and invasive neural recording studies in monkeys. And what they've found is about 20 separate areas of auditory cortex in primates. The organization of primate cortex is basically on a functional column, meaning there's cells of a column that respond to the same frequency. We also understand it's laid out in a tonotopic way. And we also recognize that there are secondary areas that do not respond well to pure tones and that have not been well researched. In other words, we have a lot of work to do to understand the auditory cortex and the auditory sensory. Making it to the auditory cortex is not the end of the connections. From the auditory cortex, there are two main streams that are conducted over to what we call the association cortex. One of those would be the prefrontal cortex, and you could see that it would be this area, and that's what we call the anterior auditory pathway. The other one would be synapsing over to the posterior auditory pathway, and we call this the posterior parietal cortex. The anterior auditory pathway, we believe, is a lot more involved in identifying the sounds as to what sound you are hearing. Having understanding of what you are listening to allows you to keep your arousal level down. If there is something that is off about that sound, it could then trigger arousal that could generate anxiety, fear, or various other emotions. Perhaps we can all agree that listening to a baby laugh is a pleasant sound. However, if we take that laughter and put it in the middle of a forest at 3 a.m. when nobody else around, that particular sound would generate a very different emotion. If you hear that, it would travel from your outer ear to the tympanic membrane. It would go through each of the ossicles, the malleus, the incus, and the stapes. It would then go over to the basilar membrane. Given that it's a high-pitched sound, it would more than likely be at the windows of the oval. And from there, it would go over to the hindbrain, over to the midbrain, MGN, over to the primary visual cortex, secondary, and from there, it would synapse in an anterior fashion, forward. The posterior auditory pathway may be more involved in locating sounds. It would actually be going this way. Where is the sound coming from? This would be important, as the evolutionary perspective says that if you're able to hear, you may be able to avoid a threat. More recently, it has been suggested that the primary function of the auditory dorsal pathway is simply just the preparation for action. Our ability to hear is not the end of the action. Once you hear and it reaches this area, the primary and secondary auditory cortex makes use of the associations it has with the rest of the brain so that you and I can take action. If there's a baby laughing in the middle of the forest, it may then prepare you to make a decision, an informed decision to run out of there or look for the baby if you'd like to give it some assistance. In addition to synapsing to the back would locate where the sound is coming from. And again, for the most part, it would be a preparation for action, survival purposes. The auditory and the visual systems interact at various points. There is evidence that suggests that the interactions between the auditory and visual systems are an indication of early and integral part of sensory processing as a whole. Synesthesia is a neurological phenomenon in which stimulation of one sensory or cognitive pathway leads to automatic involuntary experiences in a second sensory or cognitive pathway. You may have heard of this condition before, maybe not by its name, but individuals who have synesthesia sometimes claim that when they hear a particular sound, they're able to see things even though they just heard it. Or when they see the color pink, they may be able to hear a maybe high-pitched voice. And these individuals are actually looking at an interesting interaction that exists between the auditory and visual pathways. This interaction is validated when we look at the synapsing that occurs from the occipital lobe over to the parietal lobe. And we also look at the auditory pathway when we look at the primary and secondary auditory cortex. It synapses over to the parietal lobe as well, and that's what we would have as the posterior auditory pathway. Recognizing that both the auditory and visual systems are synapsing over to an association cortex of commonality allows us to understand how some individuals are able to hear things when they are seeing things, or are able to see things when they are hearing things. Efforts to characterize the effects of damage to human auditory cortex have been complicated by the fact that most human auditory cortex is in a lateral fissure. Consequently, it is rarely destroyed in its entirety, and if it is, there is almost always extensive damage to the surrounding tissue. As a result, efforts to understand the effects of auditory cortex damage have relied largely on the study of surgically placed lesions on non-humans. Given the large size of the lesions in most studies, the lack of severe permanent deficits is surprising, suggesting that the subcortical circuits serve more complex and important auditory functions than was assumed. Through these lesions that have been inferred in non-humans, mostly in rats, we see that there are very few permanent hearing deficits. The lesions that have also been done in monkeys and in humans hinder sound localization and pitch discrimination only, but not in their entirety. In fact, when we look at deafness, total deafness is rare. Even among those that are hearing impaired, only 1% is completely deaf. And we believe that this is because of the multiple pathways that we see in the auditory cortex. So you go back to the previous module, you may remember the parallel serial of understanding that this is what makes us resilient. Conductive deafness and nerve deafness are the two types of deafness that we've seen in humans. Conductive deafness would be damage to the ossicles, whereas they could have actually been destroyed, or in some cases they could have also been fused together because of unusual genetic expression that would not allow these bones to have a little bit of freedom or play between them. The nerve deafness would actually be damage to the cochlea, and this would be loss of the hair cell receptors. That is the major cause of nerve deafness. And by the way, when we expose ourselves to loud sounds on a regular basis, we are exposing ourselves to hearing loss. You may not be able to perceive that when you are young, but as we start getting to our 40s and 50s, depending on your exposure to loud sounds, you would start losing your ability to hear. For the most part, the majority of humans will lose their ability to hear the high-pitched sounds first, and then the low-pitched sounds thereafter, and that would be a natural hearing loss process. Partial cochlear damage results in the loss of hearing at particular frequencies. For example, hearing high frequencies, which is the most common. Age-related hearing loss features a specific deficit in hearing high frequencies, and that is because the elderly often have difficulty distinguishing the sounds of the letters S as in Sam, F as in Frank, and T as in Tom. They can hear people speaking to them, but they often have difficulty understanding what people are saying. Some people with nerve deafness benefit from cochlear implants. Cochlear implants basically bypass damage to the auditory hair cells by converting sounds picked up by a microphone on the patient's ear to electrical signals. These are then carried over into the cochlea by a bundle of electrodes. These signals would excite the auditory nerve. Although cochlear implants can provide major benefits, they do not restore normal hearing. The sooner the person receives a cochlear implant after becoming deaf, the more likely that he or she is to benefit, because the disuse leads to degeneration of the auditory neural pathways.